Hi friends, Namaskar. So today we are going to discuss about the analytical method validation. In the pharmaceutical industry, analytical process is very much important and that process is trustworthy which is expected by the regulatory authority. During audits or during any investigations, we need all analytical data. And so because of that, if all the data is required and most important for us, we have to do all the analytical testing validated. So for that validation, what the criteria is there, we will understand here. So core validation parameters as per ICH Q2, we will see one by one here. Specificity and selectivity, which means the ability to assess the analyte unequivocally in the presence of component, impurities, degradants and matrix. Means how much is the impurities in the particular component or the product is there, and how much is the purity which is designed or which is to be checked by this method. The accuracy means closeness of the agreement between true value and the value found, often measured by the percentage recovery. So whatever the results coming, it should be declared in the percentage and their true value to be declared and check against the specification. The precision, next is the precision. Precision means what? The closeness of agreement between a series of measurements, means repeatability, intermediate precision and reproducibility. All the results during this analytical process should be very precise. So it is called as precision. The limits or the results which is to be identified in the detection level, means detection limit, that is the LOD. Lowest amount of analyte in a sample can be detected but necessarily quantified. Okay. So basically what happened in the samples, how much is the analyte we have to be discovered with the small quantum of amount. Quantitation limit means LOQ, lowest amount of analyte that can be equivalently or quantitatively determined with suitable precision and accuracy methods. However, the linearity is ability to obtain test results directly proportional to the concentration of analyte within a given range. Suppose there is a range 95 to 105, that your result should be within that limits. And the range interval between upper and lower concentration of analyte for which the method has to be suitable, precision, accuracy and linearity. Means in this precision, accuracy and linearity, we have to discover the range of the analyte or their concentrations, upper level or lower side. The robustness, next item is the robustness, measure of the method's capacity to remain unaffected by small deliberate variations in the parameters. So this is all about the basic criteria for the analytical method validation. Now, now there is some validation steps is there. Define purpose, establish if the method is for identification, quantitative impurity testing or the assay. The protocol development, whatever the activities we are doing, it is to be protocol based, and that protocol based activities to be performed as per the criteria. The document acceptance criteria, equipment, procedures, yeah, standard operating procedure, methods, standard your STP, all these things to be required. Experimental work which conducts the studies for each parameter, that is the precision, means for which each and every parameter to be precisely discovered. Data analysis and reporting, compare the results against criteria, document and approve the final validation reports. So next is regulatory guidelines. For example, if we need to understand this process, there is a ICH Q2 R1 R2, the primary harmonized international standard. ICH means international standard for harmonization for the pharmaceutical validation. FDA and USP, specific guidance for analytical procedure within the United States, and then EMA, European Medicines Agency guideline. Methods should be revalidated. Whatever methods you are using in the quality control department, it should be validated, revalidated to have a trustworthy results. If your method is not validated, whatever the results we are getting, it should not be trustworthy and it should not give us a confidence about the product performance. So your methods should be validated. Process, instrumentation and composition of the drug product. The scope of this analytical method validation as per ICH Q2 R2 is this guideline applies to a new and revised analytical procedure used for release and stability ICH Q2 R2 guideline part 2 testing of commercial drug substances and products chemical and biological or biotechnological. These guidelines also can be applied to other analytical procedures used as a part of control strategy, that is ICH, Q8 and 10. So wherever the results are to be controlled, all the analytical testing procedures to be controlled, and the process which we are following has to be very confident, has to be trustworthy, results oriented. So we have to apply these guideline. The guideline is directed to the most common purposes of analytical procedures such as assay, potency, purity, impurity, quantitative or limit test, identity or quantitative or qualitative measurements. The analytical method validation can be measured or with respect to the impurity, purity in the assay or in the analyte, we have to understand with respect to the quantitative or qualitative mechanism. Suppose there is an analytical procedure performance characteristic to be demonstrated. So what we will going to see the type of measured product attribute. What are the attributes? Specificity, specificity test. Working range means suitability, calibration model, lower range, limit, verification. Then accuracy test and the precision. In precision there is a repeatability test and intermediate and precision test. So these all are the factors. If you want to check the impurity, that is the purity of the drug content inside the product, we have to check with the quantitative measurement. Quantitative means what? What is the quantum? Means 90%, 95%, that is when we have to calculate with the quantity basis. And assay content potency, other quantitative measurements. If we see specificity, it gives the significance or this test is normally evaluated. Impurity also evaluated, quantitative type and limit should be evaluated and assay also to be evaluated. But in the working range, identity, in the working range, suitability of the calibration model, lower range limit, verification signifies that this test is not normally evaluated. Okay. But in case of impurity, yes, quantitative detection level to be done. So limit, again this test is not evaluated. Potency, yes, it should be evaluated. Accuracy also, for the accuracy test, quantitative mechanism is normally evaluated and assay is normally evaluated. Rest test is not normally evaluated. Okay. However, the precision, repeatability test, intermediate test, here also the quantitative measurements to be evaluated, assay to be evaluated, and it should be normally significantly established. In second criteria, the reportable range of the common uses of analytical procedure. Use of the analytical procedure, low end of reportable range and high end of reportable range. Low end and high end means assay of drug substance or the finished drug product that should have a range like 80 to 120%. So 80 means the low level and 120 is the high level as per the upper specification limit. Like same in potency. Suppose if we want to say that potency to be plus minus 20%. So minus will be the lower range and plus will be the higher range. Content uniformity, like 70% declared content, that is the lower range, and 130 is the higher range. 130%, that should be a declared content, so it may be a higher range. Dissolution testing, so if 45% immediate release of the dosage form strength, first measurement time point will be your QL modified release 45%, but at the upper end it should be 130%. Same here in the impurity testing, reporting threshold or 120% of the specification limit. So this reporting threshold means it should be established limit based on the API, based on the raw materials natures, criteria and specification, as well as the pharmacopoeial acceptance level. Purity of testing is in percent, that is it should be a lower level 80% and higher level 100%. Analytical method validation, the next item we consider that multivariate analytical procedure. For multivariate analytical procedures, results are determined through a multivariate calibration model utilizing more than one input variables. Means, for example, there should be a not one variable, there should be a multiple variables to test the criteria. Example, spectrum with many wavelength variables. Means in the spectrum there should be a wavelength, but it should be a variable. So that is called the multivariate analytical procedure. Multivariate calibration model relates the input data to a value for the property of interest, that is the model output. Successful validation of a multivariate procedure should consider calibration, internal testing and validations. That is the multivariate analytical procedures. Next item is typically calibration and validation are performed in two phases. In first phase, model development consists of calibration and internal testing. Calibration data are used to create calibration model. Test data are used for the internal testing and optimization of the model. The test data could be separate set of data or a part of the calibration data set used in a rotational manner. This internal test setup is used to obtain an estimate of the model performance and to fine tune of the algorithm's parameter. Means there are certain results coming which can be considered into the algorithm way. So it has to be verified and mentioned with respect to the numerical algorithms. Example, the number of a latent variable for partial least squares to select the most suitable model within a given set of data and prerequisites. However, in the second phase of the calibration and validation is model validation and independent validation data set with independent samples is used for validation of the model. Reference analytical procedure, the samples used in validation of quantitative or qualitative multivariate procedures require should have values or categories assigned to each sample typically obtained by a validated procedure or pharmacopoeial reference procedure. When reference analytical procedure is used, its performance should match the expected performance of multivariate analytical procedure. Analysis by the reference procedure and multivariate data collection should be performed on the same sample. Means sample should not be changed during the multivariate testing within a reasonable period of time to assure samples and the measurement stability. In some cases, a correlation or conversion may be needed to provide the same unit of measurement. Any assumptions or calculations in the analytical procedure should be described in the case of multivariate data collection. Validation test methodology and evaluation is like as below. In the following chapters, experimental methodologies to evaluate the performance of an analytical procedure are described. The methodology described is grouped by the main performance and characteristic of the analytical procedure was designed for. So, in the analytical method validation, specificity and selectivity is the criteria. The specificity or selectivity of an analytical procedure can be demonstrated through absence of interference, comparison of results to an orthogonal procedure, or may be inherently given by the underlying specific principles of the analytical procedure. Some experiments can be combined with accuracy studies. So some of the cases accuracy studies is performed. Some of the cases some scientific evidences are there, scientific principles are applied for demonstrating the specificity and selectivity. Selectivity could be demonstrated when analytical procedure is not specific. However, the test for an analyte to be identified or quantified in the presence of potential interference should minimize and to prove that this test is fit for the purpose. Where one analytical procedure does not provide the sufficient discrimination, combination of two or more procedures is recommended to achieve necessary level of selectivity. Absence of interference, specificity and selectivity can be shown by demonstrating that the identification and or quantitation of an analyte is not impacted by the presence of other substances. That is means like impurity or related standard or something related substance is there. Impurities, degradation of products, related substances, matrix or other component present in the operating environment which should not impact on the quantitation of analyte or identification of the analyte. That is called the specificity. Orthogonal, the specificity can be again checked with the orthogonal procedure comparison and technology inherent justification is like that. So we will see specificity, selectivity can be verified by demonstrating that the measured result of an analyte is comparable to the measured result of second or well characterized analytical procedure, that is called the orthogonal procedure. However, in the justification, in some cases where the specificity of analytical technology can be ensured and predicted by technical parameters, that is the resolution of the isotopes in mass spectrometry, chemical shifts in the NMR signals, and no experimental study may be required unless and otherwise it is justifiable. In the case of identification, for identification test, a critical aspect is to demonstrate the capability of the identity, the analyte of interest based on the unique aspects of its molecular structure and or other specific properties. As per ICH Q2R2 guideline, the capability of an analytical procedure is to identify an analyte can be confirmed by obtaining positive results comparable to a known reference material. Means you have to take the analyte with the reference material and you should know the results of that reference material with the samples obtaining or containing the analyte along with the negative results from the sample which do not contain the analyte. Means like placebo, suppose there is not any content or the API content, so it can be a placebo. Impurities or related substances are available for assay discrimination of the analyte in the presence of impurities. Means impurities has a major role in the case of assay. If you are identifying impurity, there should be a quantification, how much impurity, what is the percentage, whether it is approved within specification, not all this criteria is there, and that should be demonstrated. Practically, this can be performed by spiking drug substance or drug product with appropriate levels of impurities and or excipients and demonstrating that the assay result is unaffected by the presence of these materials Example, the comparison with the assay results obtained on unspiked samples. So that is all about the identification. However, for an impurity test, discrimination can be established by spiking the drug substance or the drug product with appropriate levels of impurities, demonstrating the unbiased measurement. Means there should not be any bias between the results measurement or something is there. Unbiased measurement of impurities individually and from other component in the sample matrix, impurities or related substance are not available. If impurity related substance or degradation product material are unavailable, specificity can be demonstrated by comparing the test results of the samples containing typical impurities and related substance or the degradation products with a second well characterized procedure. Means here is a comparison between the test results of samples as well as the drug substance or the degradation product, which is a well characterized procedure. And so between these comparisons we can easily identify the impurity or the level of impurities in that scenario. So the pharmaceutical procedure or otherwise validated orthogonal analytical procedures should be there. Linear response. A linear response between analyte concentration and the response should be evaluated across the working range of the analytical procedure. To confirm the suitability of the procedure for the intended use, the response can be demonstrated directly on the drug substance or by dilution or the standard stock solution, which can be utilized during the analytical testing, or separate weighings of synthetic mixtures of the drug product component using the proposed procedures. What happened initially, linearity can be evaluated with a plotting of graphs, signals as a function of analyte, concentration or the content. Test results should be evaluated by appropriate statistical methods. Example, by calculation of a regression line by the method of least squares. Data derived from the regression line may help to provide mathematical estimates of the linearity. A plot of the data, the correlation coefficient or the coefficient of determination, suppose y is the intercept and the slope of regression line should be provided. In that scenario, the analysis of the deviation of actual data point from the regression line is very helpful. Means during the regression stage, the coefficient of the correlation, it is to be checked with the help of slope, that is the y-intercept, x-intercept. So data derived from the regression line may help to provide mathematical estimates of the linearity. A plot of the data, the correlation coefficient or the coefficient of determined y-intercepts with the slope of regression, that should be checked for the linearity. Means regression to be checked along with the x and y intercept with the coefficient or coefficient determination criteria. So linearity needs a minimum 5 concentrations to check the comparison or correlation between the analytes or the other chemicals impurity level. So the distributed across the range is recommended. However, the additional concentrations may be required or more complex model to evaluate the test results, because test results should be evaluated by appropriate statistical method by the calculation mechanism, and that's why the linearity should have minimum 5 concentrations to check the statistical evaluation for the test results. Non-linear response in some analytical procedure may show a non-linear response. In these cases, a model or the function which can describe the relationship between the response of the analytical procedure and the concentration is necessary. The suitability of the model should be assessed by means of non-linear regression analysis, example, coefficient of determination, or multivariate calibration. Algorithms used for the construction of multivariate calibration models can be linear or non-linear as long as the model is appropriate for establishing the relationship between the signal and the quality attribute of interest. However, the accuracy of multivariate procedure is fully dependent on multiple factors such as the distribution of calibration samples across the calibration range and the reference procedure error. There should be a calibrated range of instrument to have a strong justification or to have a strong evidence that the results are in the calibrated range. Based on signal or signal to signal noise. This approach can be only applied to analytical procedure which exhibits baseline noise. Determination of signal to noise ratio is performed by comparing the measured signals from samples with known low concentrations of analyte with those of the blank samples, means there should be a blank sample also and standard sample also. Signals in an appropriate baseline region can be instead of blank samples. The detection level and quantitation level are minimum concentration at which the analyte can be reliably detected or quantified respectively. A signal to noise ratio 3:1 is generally considered acceptable for estimating the detection limit. For quantitation limit, the ratio should be at least 10:1 is considered acceptable. In that scenario, for chromatographic procedure, the signal to noise ratio should be determined within the defined range, if possible situated equally around the place where the peak of interest would be found. The base of the standard deviation of a linear response and a slope, the detection limit, that is the DL, can be expressed DL equal to 3.3 sigma, means 3 sigma divided by S, while the quantitation limit QL can be expressed as 10 sigma divided by S. So, where sigma is the standard deviation of the response, S is the slope of calibration of the curve, the S slope can be estimated from the regression line of the analyte. Accuracy and precision can be evaluated independently, each with predefined acceptance criteria. Combining this performance characteristic is an alternative approach for evaluation of an analytical procedure, suitability described in this chapter. However, in the recommended data, accuracy should be assessed using an appropriate number of determinations. Concentration level, that is the reportable range, is 3 concentrations and 3 replicates each of the full analytical procedures. Accuracy should be reported as the mean percent recovery by the assay of a known added amount of analyte in the sample, or as a difference between the mean and the accepted true value together with the confidence intervals. An appropriate confidence interval, that is the 95%. For mean percent recovery, or the difference between the mean and the accepted true value as appropriate should be compared to the acceptance criteria to evaluate analytical procedure bias. The appropriateness of the confidence interval should be justified because it should be a confidence level 95%. Precision validations of test of assay for quantitative limit determination of impurities or purity includes an investigation of precision. Precision should be investigated using homogeneous, authentic samples or artificially prepared samples. Matrix mixtures spike with the relevant amount of analyte in questions. If homogeneous sample is not available during analysis, suppose if you do not have homogeneous sample, then artificially prepared sample or sample solution can be used. Artificially means suppose you can make a ready sample in lab and then that can be used. Repeatability should be assessed using ICH Q2R2 guideline. A minimum 9 determinations covering the reportable range of the procedure. Example 379.3 concentrations 3 replicates. Minimum 6 determinations 100% of the test concentration. Reproducibility is assessed by means of inter-laboratory trial. Means inside the lab only trials should be taken. Intra-laboratory. Investigation of reproducibility is usually not required for regulatory submission. but should be considered in cases standardization of analytical procedure, for instance, for inclusion of procedures in pharmacopoeias. Recommended data, the standard deviation, relative standard deviation, coefficient of variations, and confidence intervals should be reported for each type of precision investigated and can be compatible with the specification limit. Combined approach for accuracy and precision is an alternative to separate evaluation for accuracy and precision is to consider their total impact by assessing against a combined performance criteria, and that should be a assess with the combined performance criteria. The approach should be reflective of the individual criteria. However, it should have been established for the accuracy and precision. Data generated during the development may help to determine the best approach and refine appropriate performance criteria to which the combined accuracy and precision are compared. Combined accuracy and precision can be evaluated by use of prediction interval to assess the probability that the next reportable value falls within the acceptable range, or a tolerance interval to assess the proportion of the all future reportable values that will fall within the acceptable range. Other approaches may be acceptable if justified. The last step is that robustness, the evaluation of the analytical procedure suitability within the intended operational environment should be considered during the development phase. Means when in the development stage analysis is done, method is established, that should be tested again at the site, manufacturing site, and it should be verified, it should be confirmed good for the intended operational environment. Development phase depends on the type of procedure under study. Robustness testing should show the reliability of an analytical procedure with respect to deliberate variation in parameters. Means whatever the method validation done at the pilot level or R&D level, it should be performed set at manufacturing level, manufacturing site, and whatever the changes, analysis change, chemical change, environment change, or lab change, but all the results should be robust. That is called the robustness. Means whatever the changes is there, the actual result should not be deviated or impacted. That is called the robustness. The robustness evaluation can be submitted as a part of development data for analytical procedure or in case by case basis, or should be made available upon a request for the investigation or the future studies. So this is all about the analytical method validation. I hope you will enjoy it, go through all detail level, understood it, so that you will have increase of knowledge with respect to the analytical procedures, and it is a very crucial and important topic in the pharmaceutical. So I hope you will like it. If you like, share it, subscribe it, and comment it for the next video. Thank you.